So now it's my great pleasure to introduce today's first speaker, Madhura Day. Madhura did her master's study in India, where she specialized in biophysics and structural biology, specifically nucleic acid NMR. She then moved to Heidelberg to work in single molecule FRET on nucleosomes at the German Cancer Research Center in the lab of the late Professor Georg Langowski, who so sadly passed away a few years ago. She's now finishing her PhD with Rebecca Wade in the Heidelberg Institute of Theoretical Studies, where she is modeling single molecule FRET results. Today, she presented her latest work using single molecule FRET to study the linker DNA and the linker histone on a nucleosome. So please join me in giving a very warm welcome to Madhura, and Madhura, it's over to you.
Okay, so thank you for the very nice introduction. Today I will talk about part of my PhD project. So basically, I work with the nucleosomes. So as you have seen this very famous picture from the Olentzes in the 1970s, so they call this a particle on a string. And if you zoom into one of these particles, this is the nucleosome. So this was the first crystal structure of the nucleosome core particle from Caroline Luger. And this was a crystal structure of the nucleosome core particle with the linker histone on it. And I'm actually interested in how the linker histone is associated with the nucleosome. So a brief introduction to the linker histone. So the linker histone is about 200 amino acids long, highly positively charged. So this C-terminal domain is about 50% of the linker histone, and it's intrinsically disordered. So this one, so this actually, sorry, it's just a, yeah. So the C-terminal domain associates with either one linker DNA or with both. The globular domain is highly conserved, and based on slight amino acid sequence differences, the globular domain actually associates either with the center of the nucleosome, which is also known as on-dyad binding, or with one or the other linker DNA, which is also known as off-dyad binding. So we study this linker histone from this H10 subtype, which is an on-dyad binder. So in an on-dyad binding mode, this zone A and B, these interact with the linker DNA, and zone C interacts with the dyad. So the question is, does the linker histone have any DNA sequence preference? Now, linker histones are highly positively charged, and DNA being negative, they are very good substrates for linker histone binding. Linker histones do not have a consensus sequence because that would not make sense, because linker histones bind abundantly to DNA. But there are enough evidences that linker histones have a preference towards AT-rich DNA. The question is, does this DNA sequence preference, does it play a role in positioning the linker histone in a specific way? So in order to study this, I reconstitute the nucleosome. So it's an entirely in vitro setup. So I reconstitute a 226 base paired nucleosome with 40 base pair long linker DNA arms. I use a full length linker histone here, so about 200 amino acid long with the globular domain here. And I change the DNA sequence of these cylinders here. So I call them flank sequences because these are the sequences flanking the globular domain. So I use a single molecule FRET. And what I do is, first of all, I covalently attach fluorescent dyes onto the system. So this red dye is attached to the midpoint of this gray arm or with the midpoint of this purple arm. And the green dye is attached to the globular domain. Now, what happens in FRET is that you strike this green dye with this blue-green donor laser here. And when this red dye is absent, or if the red dye is far away from the green dye, then the green dye fluoresces. If the red dye is present and is close to the green dye, then the green dye transfers its energy to the red dye and the red dye fluoresces. Now, this transfer efficiency is inversely proportional to the distance between the two dyes. So basically, if the dyes are far away from each other, the transfer efficiency is less. And if the dyes are close to each other, the transfer efficiency is more. So you get a plot typically like this one, where a low peak would mean that the dyes are far apart, and a high FRET peak would mean that the dyes are close to each other. And this distance, I'll come to later, there's a Forster distance. This is the distance at which the FRET efficiency is half of its maximum. So you get a plot like this from here. This is the proximity ratio of the FRET efficiency. How you get the distances, it's actually a multi-step process. So it involves both single molecule experiments as well as bulk spectroscopy. You plug in all these parameters here, the Forster distance from this bulk and the proximity ratio from single molecule experiments. And from this proximity ratio data, you get a mirror image data. Basically, this is the distance. So higher FRET or higher proximity ratio would mean lesser the distance between the dyes. Lesser proximity ratio would mean higher distance between the dyes. Now, how I model it, I model not just this mean distance, but also the peak width, because it's a dynamic system. So I basically open up the nucleosome using normal mode analysis, and I screen this ensemble of structures using this software called FRET positioning and screening from Klaus Seidel's lab. And this actually gives me the theoretical FRET values and the theoretical distances. So first I used a standard non-palindromic Widom 601 sequence, where this purple flank here is GC rich, and this gray flank has 64% AT. What we see is that the globular domain exhibits higher FRET with this purple flank, which is GC rich, and lower FRET with this 64% AT rich flank. Now, this is contrary to what people saw before. So what I did as a control, so I just swapped these two sequences. So now the gray flank is GC rich, and the purple flank has 64% AT. What we see, we expected that if it actually had a preference for GC rich sequence, the gray would have a higher FRET here, the purple would have a lower FRET here, but it's not so. This is equidistant from both these arms. So this could have been a kind of an orientation of the globular domain, and this is not reflected when you swap the sequences. What we see here is that the globular domain does not show preference for AT containing sequence, which is contrary to what people saw before. But there is definitely an effect on the globular domain orientation, or probably the location. What happens when I replace this 64% AT-rich DNA with an A tract that is purely adenines on one strand and thymines on the other? Here, as you see, this gray flank has this A tract, and the purple flank is left intact, so the purple flank is GC rich. Now, it exhibits higher FRET with the A tract and lower FRET with the GC rich flank. When you swap the sequences, so now that the A tract is on the purple flank and the GC rich flank is the gray one, you get exactly the mirror image of this graph. So basically, it sits close to the A tract and far away from the GC tract. So the globular domain definitely shows a preference for A tracts. What happens when I model the distances? So this is the distance obtained from the proximity ratio. We see that there are two arginines on the globular domain that are proximal to the minor groove of the A tract. And this is the same when the sequence is swapped. So basically, these two arginines here, they are very close to the minor groove of the A tract in both these cases. So what we know so far about AT-rich DNA recognition, so this was proposed by Cui and Jurken in 2009, that in an off-diad bound linker histone, this region, this is hydrophobic, or the zone C actually, this interacts with the thymine methyl group that is exposed on the major groove side of the DNA in an off-diad. So this globular domain is off-diad, not on-diad. This was proven in silico using MD simulations by Öztürk from our lab a few years back. So what he saw was that this zone C has some valine and alanine residues that interact with the thymine methyl groups that are exposed on the major groove side. What we see is that this, on an on-diad linker histone, this zone B is involved in the A-tract recognition. So there are two arginines here that are situated very close to the A-tract minor groove. Now, this minor groove recognition by arginines is actually very well documented because A tracts, they have the narrowest minor groove and hence the highest negative charge density. And arginines are very well suited to mediate such an interaction. What we also see here is that on an on-diet position linker histone, this is unable to access the methyl groups, which are shown by blue spheres here. So these are the globular domain is unable to access the thymine methyl groups when it's situated on diet. When it's situated off diet, of course, it can interact with the thymine methyl groups here. So in order to prove that our linker histone does not recognize thymines via its methyl group, I built a special kind of nucleosome that had A tracts on both the purple and the gray arms. But the difference is that this gray A tract was complementary to thymine, and this purple A tract was complementary to deoxyuracil. The difference between these two is the methyl group. So the gray arm now has methyl groups. The purple arm does not have methyl group. So this is the proximity ratio profile from the gray arm, showing a very broad peak here that can be deconvoluted to two subpopulations. The proximity ratio profile from this purple arm actually also shows a very similar kind of plot, with which can also be deconvoluted to two subpopulations. Now I call these populations 1 and populations 2. So population 1, which shows higher fret in the thymine tract and intermediate fret in the uracil tract, would correspond to the globular domain that is oriented towards the thymine tract. And this population 2, that shows intermediate fret with the thymine tract and high fret with the uracil tract, would mean that the globular domain is oriented towards the uracil tract. Now, as you see here, so both these plots can be overlaid onto each other, and the globular domain is unable to distinguish between deoxyuracil and deoxythymine. So basically, it cannot recognize a thymine methyl group. So this brings me to the conclusion. So what we see is that the globular domain is definitely affected by the linker DNA sequence, and that it is actually oriented towards A tract. From previous work, also, if we take everything together, what we suggest that based on the binding mode of the linker histone, it can recognize AT-rich DNA via different mechanism. So when it's sitting on diet, these zone A and B, they can interact with the minor groove, and they can recognize a DNA sequence indirectly based on some physical parameters of the DNA, such as the narrow minor groove, as in the case of A tract. When it's situated off diet, it can actually directly recognize the thymine methyl groups by hydrophobic interaction. So this can actually directly recognize certain sequences. And this actually is very important biologically, because despite certain sequence difference between linker histone subtypes and despite their different binding modes, linker histones universally have an affinity towards AT-rich DNA. And that actually plays a major role in compacting heterochromatin, for instance. So, and of course, I mean, it employs more than one way of recognizing AT-rich DNA. And so this brings to my acknowledgments, actually. So I had been for 3 years in the lab of Professor Jörg Langowski and Dr. Kati Toth. And right now I'm in Professor Rebecca Wade's lab in HITS, trying to wrap up my PhD. And my thanks to my co-authors, Sebastian and Mehmet, and our collaborator, Professor Jeffrey Hayes and Dr. Cutter, who provided us with the linker histone plasmid, and my funding, my thesis committee, and thanks to all of you for listening. Thank you.
Awesome. Thank you so much. That was a fantastic talk. So we've got a couple questions in already. Mm-hmm. And just a reminder to people that if you want to ask your question by speaking it, just raise your hand and I'll unmute you. But otherwise, you can type them into the Q&A. So our first question is from Spencer Nystrom. And he asks, could an alternative interpretation of the ATGC preference be that the 3' end is always higher affinity for H1, but GC-rich 5' ends are better at interacting with H1?
So the GC-rich 5' end, you say, they are,
Yeah.
sorry, I didn't understand the question actually.
Okay, so could alternate, if the 3' end is, yeah, maybe Spencer, oh, he's got his hand raised, so he can ask it himself.
Right.
Spencer, you should be able to unmute yourself, yes.
Great, hi, yeah, sorry if that was kind of a confusing question. Maybe if you go to the slide, I think I'm just lacking the terminology to use to describe this. So if you go to your like first, when you swap the ATGC.
Yeah, sorry, I have to, okay. Yeah, maybe here or something?
Yeah, the one, wait, the one before this, I think.
Mm-hmm.
Yeah, here, right? So, wait, no, I think about this, or maybe it's the one before. I seem to recall there was one where the AT, or the GC on the gray arm was overlapping with the AT.
Right,
profile.
okay, yes.
Yeah, this one, right? So right, in your first experiment, there's a preference for the GC-rich DNA, right? But then you do the domain swap and the AT-rich and the GC are overlapping. So is that also, is the purple distribution shifting to the left on that second experiment, or is the gray distribution shifting to the right?
So basically, as you see here, the purple is shifting to the left here, which means that basically it is sitting a little further away from the purple arm than compared to this one. But
got it. I thought the gray was the one shifting, so never mind. I misinterpreted that.
Okay, yeah.
Awesome.
But
Thanks,
I mean,
Spencer.
these are different samples actually. So basically, you would have to take each of these things as individual. I mean, you can compare between this and this, but I mean, if you want to compare between, it's a bit tricky actually. These are different samples and different FRET pairs, different nucleosomes altogether.
Sure, thanks.
Yeah.
Okay, so another question. This one's from Leanne Howe, and she asks, would you predict the same linker histone sequence specificity on naked DNA?
Well, actually, this has been seen before by numerous labs. So for, as I've shown some of the literature here, so they showed linker histones are, they have a high affinity towards just naked AT-rich DNA, not just nucleosomes. Yeah.
Interesting.
Mm-hmm.
Okay. And then a question from Pallavi Mustafi. She says, wonderful talk. Hi, it's Pallavi from JNC. Hope you remember me.
Yep.
Did you check with other H1 variants, and do you expect similar kind of recognition?
So the thing is, I mean, I did not have any opportunity of working with other linker histone variants, but that's definitely a possible future direction. I mean, so I have a thing here. I don't know whether I can, that's good. So I have this kind of plot, not a plot, sorry, this table here, where you see that there are linker histones of different subtypes actually. And this arginine that we show here, it's very much conserved in all linker histone subtypes from across organisms. So basically, I think this is expected from other linker histone subtypes also. I did not do it, but that's definitely a possible future direction.
Interesting. Okay. Another question from Nicholas Sapp, who asks, could DNA flexibility play a role in linker histone binding to the AT-rich arm?
So the thing is, play a role in linker histone binding. Well, I'm not really sure about that. Actually, basically what we see from our model is that the linker histone is not really binding to the AT-rich arm only. It's still bound on diet for both the cases. It is oriented towards the AT-rich arm. But definitely, as you know, that the DNA flexibility depends upon the DNA sequence. And A tracks, they actually induce some kind of curvature to the DNA also. So in order to take all of this into account, I basically not just looked at one, so I showed you one model, but I modeled an ensemble with different arm flexibilities also. And this actually describes not just the mean, but the peak width of my FRET data. So yeah, but whether the linker histone flexibility only is responsible, I'm not really sure of that.
Awesome. Yeah. Okay, I think we're going to wrap up there. And I'll note that there are many more questions in the chat and some raised hands that I've missed. So if your question wasn't answered, I'd encourage you to stay for the coffee chat afterwards, and there'll be lots of time for extended discussion. But let me thank you again, Madhura, for the fantastic talk.
Thank you. Thank you.